In this figure, similar figure before, so Punnett square is going to summarize meiosis and fertilization. So here are two alleles, big G, little g, little g. So you have two alleles, same gene, that means two different versions. And when they go through meiosis, these are the possible gametes. There can be a gamete with a big G, another one with little g, but because you get 4, it can be 50-50. And then same thing here, there's only one possible version of the gene here for the second parent. Goes through meiosis, the only possibility there is the big G. But then fertilization happens. Let's say this is sperm cell and this is egg cell, fertilization happens. These are the possibilities for the offspring. It is possible for this one, it's only big G that's gonna go, either if it's a sperm, then that's the only one that can fertilize. Here you have two different possibilities. So depending on if this is going to fertilize this egg, you're gonna have big G, little g. If it's fertilizing this egg, then it's gonna be big G, big G. So this is again independent assortment. There is a probability, equal probability that you're going to get this and then you're going to get this. So it depends again completely on independent assortment, and it happens randomly. This is known as law of segregation. It's going to segregate the alleles into gametes through meiosis. So this is Mendel's law of segregation. So you can create a Punnett square taking this information, and then you can look at the percentages. Here's for humans, I think Kunal asked that question today. So in humans, we can use Punnett square to track the inheritance of genetic disorders. This one is showing cystic fibrosis. In this one, mother is a healthy carrier. That means she's not sick, she's carrying the gene for it. So letter F is maybe affected gene. Father is healthy carrier as well. So he's also carrying the gene for it, but he's not sick. When they reproduce, they're going to have big F, so healthy non-carrier, big F, little f, healthy carrier, big F, little f, healthy carrier, and then affected, little f, little f. Then you look at the genotypes, you have one homozygous dominant, so it's 1:1 ratio for genotypes. For phenotypes, you have one who's healthy, 2 that are carriers, and 1 who's sick, affected, 25% chance. So if you go to the doctor or the genetic counselor and ask them, what is the chance my child's going to be sick? They might do the Punnett square depending on your gametes, and they're gonna say, oh, it's 25% chance that your child might be sick with the disorder. So that's how they can use to look at the disorders. I think I might have a question for you guys. So there's a question. I want you to do the Punnett square, and I want you to tell me the answer to this question. Now pause the recording. So I'm gonna give you a minute to do this. Okay, so yes, the answer is C. I think it's the same Punnett square. So you have healthy, big F, little f, and then the affected, little f, little f. No, this is a different one. So in this case, you had big F, little f, and then you had little f, little f. So that would give you 50% chance for that. I also want to show you one more thing since we are on genetic disorders. This is called pedigree analysis. This is a royal family of England. So this is like your family tree. You could do this one for your own family. So here the red is showing you affected individuals with hemophilia. Have you guys heard about hemophilia? Do you guys know what hemophilia is? No?
You're talking blood disease or blood disorder?
The blood disorder, that means you just can't. So a normal person, when they get a cut, there's a clotting proteins, they come in, they clot, and then the bleeding stops. Person who has the hemophilia disorder, they can't stop bleeding basically. So their body doesn't make those clotting proteins. So royal family of England, they had, so Queen Victoria, she was a carrier for this. So she was affected, but not all the way basically. So it still caused some symptoms of bleeding. So when they had children, these are the chances if either they had full-blown hemophilia or if they were carriers. So circles are representing females and the squares are males, and you can see that females are mostly carriers, and then the males, they are full-blown affected. Another thing that happens every month for ladies is menstruation. So if you have this, if you're a carrier for it, there's a chance that you're going to bleed a lot. So one solution of Queen Victoria to this was staying pregnant. So look at the number of children she had. So that was one of her solutions that she did. Another thing that we've been talking about is inbreeding. So royal family people, they don't marry commoners. They didn't marry commoners, at least. And I don't think this one is up to date. So what they would do, they would, either marry other royal families, or maybe they're, far cousins in royal families. And when you're inbreeding, so even marrying your cousin is inbreeding, that's increasing the chances of the recessive alleles. And if you're a carrier for it, there's a higher chance that if you're inbreeding with a person who's also a carrier, you're going to have a full-blown disease. You're going to have, your offspring is going to have the disease. So that's why inbreeding can increase the chances of the genetic disorders. But this is called the pedigree analysis. I have another question for you guys. When they do the gender reveal, so nowadays that's a thing, I guess. What is the chance the child is going to be a girl or a boy?
50.
50. Do you know how you get the 50%?
Yeah,
So
heard this before. There's some. It's not exact.
it's not exact. So let's do some math here. So this is a symbol for males. I will expect you guys to know this. What is the sex chromosome for males?
YX.
XY or YX. And then this is a symbol for females. What about their sex chromosomes? What do they have?
XX.
X.
And then this goes through meiosis. And so this is diploid. And it goes through meiosis. Now for males, there might be a sperm that has X, and there can be a sperm that can have Y. So these are haploid. And for females, it can be X. And the other one is also X, but I'm just gonna write it out. So there's only one possibility, basically, that's X. So I have only one version, but I just wrote them down both. So this is haploid, these are the gametes. So then you go ahead and do the Punnett square for that. So I'm gonna do a Punnett square here. So for females, you have XX. And for males, you have XY. So you bring this X down, another one from this side, same thing here. This one comes down, and then this one comes here. And then X down, and then here you have Y, X, and then Y. Now look, when you look at the genotypes, so you have 50%, it's gonna be XX, and then you have 50%, it's gonna be XY. So it's 50%, it's going to be female, and then 50% male. Does that make sense?
Yes.
Okay, so I want to know if there is someone with a big family, if you have lots of sisters and brothers. Anybody?
I do.
I do too.
How many sisters or brothers? Catherine?
2 sisters and 2 adopted brothers.
Okay, what about the other person? Who was the other person? Who said?
It was me.
Lawrence?
Um,
Okay, Fatima?
Oh, sorry.
Go ahead.
I'm sorry for the noises. We're 5 sisters, 2 brothers.
So Fatima said it's 5 sisters. So 5 sisters and 2 brothers. So using Fatima's family as an example, I want to ask you guys. So this is not 50-50. I want to ask if this is violating this. Is this violating this rule here, 50-50?
Yes.
Yes.
Chris said no. Why no, Chris?
This is Justin. I think it's no also.
Yes, it's a no. And the reason
It's
is
so
because
random.
this is the probability of a single child. We're not looking at all the children together. Usually students make this mistake when this question comes up. This is for a single child. There is a 50% chance this child is going to be male. This is a 50% chance the child is going to be female. So this is for each child. So all the 5 sisters have to be considered differently. And then same thing here. So 7 children, but for each of those 7 children, the chance is 50-50. Does that make sense?
Yes.
Yes?
Yes.
Okay. Another thing I want to mention, last time I mentioned that these insurance companies, they collect data and there is a high mortality rate for males versus females. Also, when they collect data on birth, there is a higher chance of males versus females. So more boys are born compared to girls. And the reason there's still a lot of debate on it. They said that Y chromosome, it's lighter in weight. So therefore the sperm that carries the Y chromosome swims faster to the egg. That's one of their explanations, but there's a lot of debate on that. But that also kind of makes up, because there's higher number of boys born, that also kind of makes up them, the death rate of boys there, because males have higher death rate compared to females, younger males. So that kind of makes up for that. So I want to know if there are any questions. So this one is like flipping a coin. Each time you don't know what you're gonna get. It's either head or tails. That's what, for each child. So that's like 50-50. So this does not violate this rule, because again, this is for a single child, single individual or offspring. XL, no. We're talking about the sex chromosomes here, so we're not talking about dominant and recessive here. And next time we're going to talk about, there are some sex-linked disease. That means they're linked to the sex chromosomes. So that's a little bit different. So I want to show you the dihybrid cross before I end today. I don't want to start the dihybrid cross because I want you to practice with monohybrid first. So please do the practice worksheet. But I do have some, I want to just give you, actually there's a question. We did that. So this is what the dihybrid cross looks like. Now you have 2 genes you're looking at. So this is 2 genes on different chromosomes can be combined into one large Punnett square. That's called dihybrid cross. And look how complicated it got. You just added 1 more gene. I'm not sure what their the letters are for, but it can be maybe the color and the height. Or actually here, it's wrinkled and green, wrinkled or yellow. So they're basically adding another gene. One was for color. They're adding another one if it's wrinkled or not, wrinkled or round. So after adding another gene, it just gets really, really complicated. So we're gonna talk about this next.